joining today's event. This is the first time we've done this type of event. Instead of a traditional workshop with presentations, we have 3 breakout rooms operating at once on 3 different topics. We have guide RNA design, vector cloning, and plant transformation. And I'd like to introduce today's speakers very briefly. So speaking on guide RNA design is Kobe Stalker and Eric Myers from the University of Minnesota, and Vina from the Donald Danforth Plant Science Center. In the second breakout room covering vector cloning is Josh Clem from the University of Maryland and Keunsub Lee from Iowa State University. Lastly, Wayne Parrott from the University of Georgia is covering plant transformation. I'm so grateful to all of our speakers that they're here to share their expertise with us. I'm sure you feel the same. So if you'd like to learn more, you can visit our website and read their biographies. So in a minute here, I'll open the breakout rooms. You are encouraged to move between the different rooms so that you get information on all 3 topics. You should be able to exit a room and come back to the main room, or you can move directly between the 3 breakout rooms. Once you are in a breakout room, you can either raise your hand to ask a question using the toolbar at the bottom. There's a raise the hand function. So you can raise your hand to ask a question, or you can type your question in the chat. All of the breakout rooms will be recorded and available online.
Okay, so I'm interested about the design in contract for CRISPR editing purpose. Specifically, can you tell us about, for example, for targeted TDA, targeted insertion design for SDN2 or SDN3 purpose, not for SDN1 knockout, any suggestion from you? So when we design the contract, particularly, to deliver the DNA repair templates.
Right. So that's a good question. So I don't actually work very much on HDR, or homology-directed repair, which is what you're referring to, or site-specific insertion. There are various parameters that I think have been tested in plants, including and not limited to size of the homology arms on your donor template, the methods in which you actually deliver the donor template. However, I'm not sure I could provide like a very detailed explanation on how to best design an HDR experiment itself, unless maybe there's any more specific question regarding it.
So Hong, were you referring to HDR, or were you referring to just site-specific integration?
Well, so for example, generally, when we design and CRISPR contract, we need to consider, the expression of nucleus guides, and also because if we think about HDR editing, immediate editing, we need to consider the delivery of expression of DNA repair templates as well, right?
Yeah.
Something, if you have experience.
Right. So I don't have too much experience in doing that. However, most of the time, the template itself is not delivered on tDNA. However, it can be delivered on tDNA. Much of the time, the template is delivered through a bombardment or ballistic bombardment of the template itself, whereas the nuclease itself can be delivered on a separate plasmid along with the template. So it would be like a co-bombardment strategy. However, you could deliver it on tDNA. It's less frequently used, in which you deliver both the nuclease machinery and the donor template. However, it's worth noting that we view HDR sort of as the gold standard in plant biotechnology, and specifically in CRISPR technology. And it's something that we're working really hard on improving the efficiency for, but it sort of lags behind the other modalities of genome editing. It's very challenging to do, and because it seems to be so locus specific, delivery strategy specific, and species specific, it's difficult to provide a set of directions or parameters that can be used to best achieve HDR.
Any other questions?
Oh. Thanks.
Yeah. Okay. I don't see anyone raise the hands here, but you can turn on your mic and then ask questions.
Looks like we might have one from Lucas.
Oh, hi, can you listen to me?
Yep.
Yes.
Okay, so I have a doubt about assembly of vectors with multiple gRNAs for multiplex gene editing.
Sure.
So I think there are a few systems that use golden gate cloning to assemble like a CRISPR vector with multiple gRNAs spaced with either tRNAs or CSI4
Sure, yeah.
sites. And I was trying to use such systems, but I had a lot of difficulty trying to clone vectors with 6 or 4 gRNAs to edit like 3 or 2 genes simultaneously, because the cloning efficiency seems to like drop quite a bit as the number of fragments in the reaction increase. Although the protocols state that is perfectly possible. So I could clone like vectors with 2 gRNAs, no problem. But when I try to up the amount of gRNAs, I tried lots of things like changing the typing parameters and enzymes quantities and all. But I had a lot of like pretty much false positives and I had like to I did manage to get a vector for with 4 gRNAs, but it was like I had to like to sequence a lot of colonies. So I'd like to know
Yeah.
if you guys like face the same difficulties and if you have any suggestions to what kind of system to use
Yeah.
and what other strategies I could use for monocots specifically.
For monocuts, sure. So specifically, I can refer you to a system that we use, which has been tested in many different labs and used pretty broadly within our field. So we use a Golden Gate strategy to assemble up to 8 nucleotides, but we also have, 8 guide RNAs, but we also have a strategy to assemble up to 16, which is done a little bit differently. So I'll just discuss the one in which we use up to 8. And I actually maybe can share a slide, so I'm not just trying to explain it with words. It might be easier to have a visual aid. So I'll
That
just.
would be great, actually.
Okay. Are you able to see my PowerPoint here?
Yes.
Yeah.
Okay. So basically, in our system, so this is the original publication in which we produced this system, and we've adapted almost all of our reagents to be compatible with this cloning strategy. So all of our reagents for base editing, for transcriptional regulation, and for mutagenesis with different nucleases like Cas9 and Cas12a are compatible with this strategy from this 2015 article in Plant Physiology. So essentially, let's say, for example, that we're interested in assembling a tDNA to express 4 different guide RNAs, maybe using Cas9 as our genome editor. Our first step would be to design oligos that are complementary to each other, which are also specific to the guide RNA. And we would digest a backbone vector in which, if we're using 4 different guide RNAs, we digest 4 different backbone vectors. So PYPQ 131, 132, 133, 134. And in this case, we're processing each guide RNA with using a tRNA-based strategy, right? And there's a few reasons that we use the tRNA strategy. I know you had mentioned CSY4. We prefer this over CSY4 because it doesn't require the expression of an additional protein, which would be CSY4, of course. And we can express multiple guide RNAs. If you look at the final construct, for example, here at the bottom, under a polymerase II promoter, with each guide RNA being flanked by a tRNA RNA sequence. So basically, we would anneal the guide RNAs after digestion of these backbones using BSMB1, and we would ligate it into that backbone, and then we perform a Golden Gate reaction based on the sort of color-coded sequences here. and using Golden Gate with BSA1, combine that all into a Golden Gate acceptor clone, which harbors that polymerase II promoter that I mentioned. In this case, we're using ZMUBI. And this reaction in particular is very efficient. So as I said before, I've assembled up to 8 using this system. And while there is some reduction in cloning efficiency as you scale up the number of guide RNAs, it seems to be efficient in the sense that you don't have to screen. You can really screen 1 or 2 colonies, right? And we also couple this with blue-white screening. So we select the white colonies, and it's not uncommon at all to only have, say, maybe 1 or 2 blue colonies on a plate full of colonies. So I would certainly recommend looking into this system in particular if you're interested in expressing multiple guide RNAs under a polymerase II specific promoter. This one works very well, and we also have different compatible vectors that aren't using the tRNA processing strategy. For example, we also make use of the Hammerhead HDV ribozyme processing strategy, where instead of a tRNA, each guide RNA is flanked at the 5' end by a Hammerhead ribozyme, and at the 3' end by HDV ribozyme. Okay, so that's just another option that we use. But our lab, and I'm in Yiping Chi's lab. Maybe I should introduce myself actually in a moment just to let people know what my background is. But our lab for monocots in particular most often uses this strategy using a maize ubiquitin promoter for expression in monocots and multiple guide RNAs being flanked by the tRNA sequences. Hopefully that answers your question, Lucas.
Yeah, for sure. Thank you very much. I will take a look at the article, but I would just like to clarify one thing.
Sure.
So in this system, each gRNA is first cloned into a backbone before a different backbone. To explain, the system I was using was
Exactly.
a system like developed in Voita's lab, it was the CERMAC 2017 article, and in this system, each gRNA is the overlaps for the Golden Gate are like in the middle of each gRNA between bases 8 and 12, if that makes any sense.
Yeah.
And from what you explained, it seems to me that you have like restriction sites outside the tRNA gRNA complex. Is that correct?
Yes. Yeah. So I tried to color code it in a way that it would be clear, but the yellow sequence, when it digests, would ligate into the backbone acceptor, which is PYPQ144. And then we have these 2 red sequences ligating or annealing rather. The green sequence is annealing, and the blue sequence here annealing. And then the final guide RNA is flanked with the gray sequence, which ligates also into the backbone.
Oh, perfect. So that the reaction uses a single restriction enzyme. I assume.
That's correct. This is done as a typical Golden Gate reaction with where it's all in one pot, right?
Oh, perfect.
Using BSA1. Typically we use BSA1 high fidelity, which you can purchase from New England Biolabs.
For sure. Yeah, because I think the system I was using uses 2 enzymes in the reaction, and that's one of the things I assumed could be causing some trouble. But anyway, thank you very, very much.
Right. Yeah. No, not 2 restriction enzymes, but there is an additional T4 ligase, right? So there are 2 enzymes, but it's just BSA1 and T4 DNA ligase.
Okay. Thank you, Josh.
Awesome. Thank you.
Here's the next question. Is it necessary for each gene in a multi-gene construct to be regulated by a different promoter and terminator to prevent transcriptional silencing?
So in the ideal sense, I think that would be the case, and not just for transcriptional silencing, but also to avoid plasmid recombination within Agrobacterium. Because if you think of the strains that we often replicate plasmids in, such as DH5 alpha, those are recombinase mutants, and so you're unlikely to observe any form of plasmid structural rearrangement or recombination. However, when you take that out and you're ready to transform your plant, you put it into Agrobacterium. Those are more susceptible, say, to plasmid rearrangements. And that would be one reason to avoid using so many repetitive sequences, right? In fact, that might be one of the main reasons you would be interested in avoiding repetitive sequences. So what I'll say is that ideally, yes, you would want to avoid using, say, multiple copies of the same promoter and terminator set. However, most labs, including our lab, don't really follow that rule perfectly. It's very common for us to use multiple maze ubiquitin promoters and multiple NOS-T terminators. I wouldn't want to exceed 3 of them. But one way that you could avoid uh, gene silencing is to avoid using non-native sequences or non-plant derived sequences, so sequences from viruses or even from bacteria. You may be avoiding something like 35S or the NOS-T terminator. If you're really concerned with transcriptional silencing, that would be one step to take to avoid using those non-plant derived sequences.
Thank you. The next question from Joy Joey is, what is the criteria for selecting guides after analyzing secondary structures?
Criteria for selecting guide RNAs. So this is sort of difficult because there's no perfect parameter. There is no such thing as a perfect guide RNA, right? I think a lot of it depends on the locus at which you're interested in performing genome editing. In particular, you have aspects such as the chromatin accessibility, which we know has an influence on the efficiency of a particular guide RNA. Some parameters that I personally use, and by the way, I design almost all of my guide RNAs manually. I know that there are softwares out there that will help you to do it, but I use a few basic parameters. I try to keep the GC content between 40 to 80%. Ideally, I want to be around the 50 to 60% range. Okay. I also avoid highly repetitive uh, uh, polynucleotide sequences. So I wouldn't want to have, say, like 4 Gs in a row for a number of reasons. Probably not ideal for secondary structure, but also for synthesis reasons. Sometimes if you're synthesizing guide RNAs with multiple copies of the same nucleotide in a string, you have some synthesis problems and you might not recognize it until later on. Finally, I like to include things that you would also include in like primer design, for example, such as a GC clamp. I prefer to have a G or G's and C's kind of at the termini of my guide RNAs, just for better, perhaps better annealing based on the 3 hydrogen bonds between G's and C's, right? So those are a few parameters that I include, and then also off-target sequences, something to consider as well. However, off-targeting can be very challenging to avoid. I wouldn't want to design a guide RNA that has a perfect match to an off-target sequence within the genome. I will tolerate some mismatches because those get very difficult to avoid. And also when you start to include mismatches in your guide RNA, the efficiency tends to go down a lot. But I've covered some of the parameters of guide RNAs, although there is a breakout room, by the way, specific to guide RNA design if you're interested in having more of your questions answered on that. So hopefully I've addressed what you've asked.
Thank you. Does anyone have other questions?
Hello, sir. I'm Abhinav Tiwari, and I'm a PhD student. I'm doing PhD in plant pathology from Asha Narendra Agriculture University. Uh, I actually have worked in plant viruses, so I've gone through this vector cloning and guide RNA somewhat. So I'm very interested to know this whole process properly. So can I get this recording, because I've joined this in between? I was attending the guide RNA class earlier.
Yeah, so the recordings will be available after meeting.
Yeah.
Okay,
Yeah, it'll
thank you
be
so much.
posted to the Plantin website.
Sure.
Okay. Yeah, I think another thing you can consider when you selecting guide RNAs is the promoters you are actually using for guide RNA expressions. Whether
Yeah.
you're using like U3 promoter and U6 promoters or polymerase 2 promoters, as Josh just mentioned, then your cloning strategy will be a little bit different.
Yeah, thanks, Keunsub. I actually neglected to mention that in my response to the last question. So we typically express them in two different ways, right? Under either a polymerase 2 promoter, which I've depicted here, maize ubiquitin, or a small RNA polymerase 3 promoter. Those are either U3 or U6. So either way, what we typically use polymerase 3 promoters and dicots, by the way. Just historically, that's how we've done it. They're also very effective in monocots, though, so nothing wrong with that. They're typically terminated by poly T, so multiple T's in a row signals. So they don't require a polymerase 2 terminator. And then other design parameters, if you do opt for pull 3 or pull 3 U6 or U3 promoters, which we have compatible with our vector system. I might have a slide on that. I might need to check. If you're using polymerase 3, the first nucleotide of your guide RNA needs to begin with an adenine because that's the transcription start site for polymerase 3 promoters. If you're using polymerase 3 U6, if you're using the U6 promoter, it needs to begin with a guanine because that's the transcription start site for U6. So those are two important design aspects if you do opt for the polymerase 3 promoters. Yeah, see if
Josh, here is next question. Do
Sure.
ubiquitin promoters from maize work well in dicot for Cas12 and the guide RNA expression?
So I don't know the exact answer to that, but typically we avoid using, say, monocot promoters in dicot species. We try to keep it species compatible. So for a dicot, our system uses Arabidopsis ubiquitin as the promoter, and for monocots we use maize ubiquitin. We don't interchange them for dicots and monocots. We stick with that, and that's, I think, just a general rule if you're interested in achieving strong constitutive gene expression in either a monocot or dicot species.
Which strategy would you use to change promoters driving Cas12 expression to native promoters?
So I guess it would depend on the species that you're interested in expressing Cas12. As mentioned, we really just drive Cas12, at least in monocots, with polymerase 2 maize ubiquitin, or in dicots with an Arabidopsis ubiquitin promoter. Now, I will grant that perhaps using the strong ubiquitously expressed promoters for Arabidopsis genome editing might not be the best way to go about it if you're interested in reducing the level of mosaicism. So it's often it's often that researchers will opt for, say, an egg cell specific promoter to drive their nuclease for genome editing in Arabidopsis, and I think that's probably a good thing to do because that tends to in that first generation increase the frequency of biallelic mutations, right? So I think EC 1.2, an egg cell promoter, PD D45 is another egg cell specific promoter. That's specifically for Arabidopsis. Driving Cas12a in a monocot, I would just use probably something like a maize ubiquitin promoter, which is what we do.
Okay, thank you, Josh. Any additional questions? Please unmute yourself and speak. Okay, here's the next question.
I need that.
Okay.
No, I wait.
Okay, I will just read the question from home again. Could you please tell us more about your experience with the ribozyme cRNA expression systems How efficient is this system in monocot and dicot plants, respectively?
Sure. So typically we actually only use the hammerhead HDV ribozyme strategy for expression in monocots under a polymerase II specific promoter. They do work in dicots. We just historically have been using them in monocots. And from what we know about the hammerhead HDV strategy is that it precisely excises because it's a ribozyme, right? So it doesn't require any other endogenous machinery for excision. For example, the tRNA strategy requires endogenous RNases to cleave them out and process the guide RNAs. However, with hammerhead and HDV, they're ribozymes, so they are self-processing. And the hammerhead ribozyme precisely cleaves at the 5' position of the guide. And the HDV ribozyme precisely cleaves at the 3' end of the guide. So that's why we put hammerhead on the 5' end and HDV on the 3' end of the guide RNA. Now, we specifically use that system for Cas12a because the stem loop, or you might think of it as a tracer RNA scaffold or feature of the Cas12a is at the 5' end of the guide. Okay, and that hammerhead actually requires a 6 nucleotide complement at the 5' end. And so if you're using that for Cas9, your guide RNA begins with your unique sequence, and then you would have to change the sequence of your hammerhead to complement with that for every different guide RNA that you use. So using the hammerhead HDV strategy for multiplex genome editing with Cas9 can be needlessly complicated when we have a tRNA processing strategy that you don't need to adjust any other parameters about the tRNA sequences themselves. For Cas12a, you don't have to worry about it because the stem loop is at the 5' end of the guide and it remains the same. Okay, so you can use the hammerhead without modifying it every time. However, I've used it and our lab uses it quite a lot. The hammerhead HDV strategy results in very precise and efficient processing of individual CRISPR RNAs.
Thanks for that explanation. So one more question related to this. For multiplex editing, how many guides can you use same time?
Yeah,
using this system.
Sure, we've done up to 20, even with Cas12a. We've done up to 20 new guide RNAs using a. I will go to the slide. Using a higher order assembly. Actually, this is showing 16 guide RNAs, but they edited, I think, up to over 20 target sites with this particular vector. This is accompanied with the paper published in Nature Communications on multiplex editing using Cas12a, but we achieved a high frequency of biallelic genome editing using up to 16 guide RNAs and this guide RNA processing strategy. So that to answer your question, our lab has done up to 16. There may be reports of more than that that I'm not familiar with.
Okay. Aileen, please ask your question.
Great, thanks.
Sure.
Hi. Sorry, I'm late to the meeting, and I want to ask you about the use of CS4 for mono. I know that in barley and some mono is toxic, have a toxicity. Do you know if the use of in rice, for example, have a kind of toxicity too?
Are you referring to CSY4? I couldn't quite hear.
Yes. CS, the protein that the CACE4 protein to enhance the guides.
Okay, so yes, CSY4 I think is the one we're talking about. So
yes,
we've used that in rice and at least haven't observed any problem with toxicity. That's not something that we've observed. However, I will say we don't really use that system at all anymore in our lab.
Okay.
Now, I personally am experienced using a type 1 CRISPR system in which we use CSY4 as part of the cascade because it forms a complex upon
Yes.
genome editing. And I express CSY4 as part of that complex. It's also Cas6. They're interchangeable, Cas6 and CSY4. I don't experience any toxicity in expressing that cassette in rice, at least.
Okay.
So that would
Because
be, I don't use it for guide RNA processing, but that would be at least some evidence that it's not toxic in rice.
okay, because we use it for wheat, for example, and we don't have problems. In barley, we observe toxicity, and nowadays we start to work with rice, and well, we want to know if have the same problem with barley.
Sure, yeah. So no problems in rice, but I can't really say much about other species.
Lucas, please unmute yourself.
Okay, so since Ren asked another question about multiple gene editing, I actually have another question. Uh, is there much of a difference between using like a polycistronic approach with tRNA spacers, which is the one actually I was trying to use, and using multiple transcripts, each one with a promoter and a gRNA? Because that's currently the alternative I was going with because I had a lot of difficulty on the polycistronic approach.
Sure. So what I would say, if you're having difficulty with the polycistronic mRNA approach, I would say test out the polymerase 3 promoter approach because there's nothing wrong with using that system per se. There are a few potential advantages of using POL2, right? One of them being that you have coordinated gene expression between uh, all of your guide RNAs, essentially, because they're all driven by the same polymerase machinery. So you get equal coordinated gene expression through all of them. And there are also some mechanisms by which that RNA might be protected, with the poly A tails. So you might achieve greater gene expression using of the guide RNAs using a pull-2 promoter. Additionally, specific to the tRNA sequences, it turns out that they have some polymerase 3 activity, so they can actually recruit polymerase 3 to them as well, which can boost your gene expression per se, right? So those are a few advantages of using the tRNA strategy with the pull-2 promoter. However, as mentioned, if it's not working for you well, I would test out the polymerase 3 strategy. Just make sure you design your guide RNAs adequately with the U3, U6, beginning with adenine and guanine, respectively, as you're designing your guide RNAs. So that would be, I guess, what I have to say there. Hopefully that addressed your question.
Yes, thank you very much. I didn't know that about the tRNA recruiting the polymerase.
Yeah.
And just one extra thing, because you've mentioned that like you had efficiencies such as like you only needed to screen for 1 or 2 colonies, and that like almost made me cry because I had to screen for up to 30 colonies.
Yeah.
And I was wondering, one thing that was very common to me when screening colonies is that I noticed that um, when sequencing those colonies, that we had like vectors that would like lose the CCDB site and close in the edges. Is that common? Does that happen to you?
So we only include a CCDB containing vector during our gateway step, not during our Golden Gate step.
Oh, yeah, okay.
So in Golden Gate, we have LAC-Z, which enables blue-white screening. Do you have a LAC-Z gene?
No, it's only CCDB for selection.
Okay. So that is one difference, at least with our system, is that we're using LAC-Z, which enables really nice visual screening. However, it's not uncommon for me, at least, to not even do the IPTG and XGAL step because it's so efficient that I don't really need to. But you can if you're interested in doing that. And then we include the CCDB for this multi-site gateway reaction where we include a destination vector harboring the tDNA backbone, right? Sorry. the nuclease vector, and the Golden Gate product guide RNA vector. Okay. So basically the Golden Gate product goes into our gateway reaction after it's done.
Okay, yeah, I'll take a look at that system. In this system, you have to have like one backbone for each gRNA, right? Because I was wondering, because here in Brazil it's quite expensive to import the backbones.
Yeah, sorry to hear that. That is one downside, and another downside that I'll admit about using this system is that the LR clonase 2 is a very expensive master mix or enzyme mix, right? So if you're working with another system that just uses Golden Gate, you might run into some of the issues that you're mentioning, because the Golden Gate plasmids that we put in, the entry clones and the destination clone, are much smaller than what our final tDNA vector is. So that could be, part of the reason that you're observing some low reduced efficiency compared to what we see. But yes, they are all individual backbones. Yeah.
Thank you very much.
Thank you, Josh. Who else has any questions? Yeah, just feel free to unmute yourself or raise your hand or type the question into the chat. Josh, looks like you have a lot of experience with different.
I have one more question.
Sure,
Regarding the promoter or terminator we use to drive is a nucleus or guide expression, right? So can you tell us more about the terminators? How crucial they are if we consider design contract.
Sure.
Of course, we need to consider the promoter, right? But the terminator as well. How crucial compared to the promoter?
Yeah. Actually, my figure is missing a terminator, which I didn't include for simplicity of the figure, but we have, of course, terminators at the end of our Cas9, and usually just a nobuline synthase terminator, NOS-T. Now, there are definitely stronger terminators out there. And I think that's a relatively unexplored kind of optimizing terminator sequences for increasing gene expression, especially related to CRISPR constructs. For example, I know that there's another terminator out there that's from Arabidopsis. I think it's heat shock 18.2 terminator that is documented at least to increase gene expression relative to some other more commonly used like NOS-T terminators. And that's one way in which you could likely improve CRISPR-based genome editing just by sort of optimizing the expression of preventing transcript read-through, right, would be one goal with that. Also reducing silencing, since Arabidopsis heat shock 18.2 as a terminator is derived from a plant, could reduce silencing. So I will say we just use NOS-T most of the time, but it's something that could be improved on.
I see. Do you recommendation, for monoclonal, diclonal, the best, potential terminator we can use, if we do not consider NOS terminator?
I think another one. I don't do much genome editing in dicots myself, actually. I think the 35S terminator would be an adequate one. Maybe Keunsub, do you have any recommendation for that?
Yeah, actually, if you want to optimize it, you want to test the different combinations of the promoter and terminator interactions.
Yeah.
If you're looking for a stronger expression, extension gene promote terminator from tobacco gives a very, very strong expression in both monocot and dicot. And you can also consider chimeric terminator or 3' UTR. So this one already published several years ago. You can look for the chimeric 3' UTR for gene expression. It was a 2018 Clem Biotech Journal paper.
Oh, okay.
Yeah, they tested, nose terminator was the control, and they compared the 35 terminator and the chimeric terminators plus matrix attachment region, or MAR. So if you really need a higher expression, you can consider those terminators. However, it was validated in Benthamiana, right? So it doesn't mean it'll work equally well in your own species of interest.
I see. So that means, when we consider, let's say, different promoter, different terminator, we also need to consider combination, right? Both to
Yep,
get
it's in
really the
combination.
maximum expression. I see.
Yep.
Got
Okay,
it. Thanks.
now, we have a next question. Where can we get this multiple gene editing gateway vector? Can we use it in Poland, a polyploid like the banana?
Certainly, actually, we've had collaborators that have had successful genome editing in banana, using our vectors. I don't know because I'm not involved in that project. I don't know all of the details on it. I think your question is where we can get the vectors, and they are all deposited to AdGene. So we have over 300 vectors on AdGene. Sorry, I'm just going to skip through my slides to get to the slides that have that information. And I also had some slides on the guide RNA processing. So if you go to our lab page on AdGene, Yiping Qi, you can find, you can also search based on the publication. So the publication I flashed earlier, that from 2015, published in Plant Physiology, you can look at all the vectors coming from that particular article and find the associated plasmid with it. I know the multiplex guide RNA entry clones are PYPQ131. For the polymerase 3 promoter containing vectors, it's 131 A or 131B for Arabidopsis, and I believe it's for rice U3 and U6, it's 131C and 131D. You can double check that information though through AdGene here. It's all on there. We also have the ones containing tRNAs and the ones containing the hammerhead HDV ribozymes.
Okay, great. I think we have a few more minutes. Yeah. Lucas, are you still
Actually, I have another question. Sorry if it's too many. But you mentioned that you did up to 16 gRNAs in your lab. That was quite impressive. And I was wondering, like, in those cases, what kind of strategies do you use to screen for plants that are edited for all of those sites and potentially homozygous for all of them? Because I imagine like a lot of work.
It is, yeah. There are probably faster ways, but we opt for, because we're often screening a lot of plants, and we're also interested in assessing the editing efficiency at the molecular level, right? Not just based on phenotype. We're interested in looking at the zygosity, the level of mosaicism or chimerism. And we can best do that and most efficiently cost-wise using amplicon deep sequencing. Okay, so we send our amplicons for Illumina sequencing. Let's say we have 20 different plants, and let's say we're looking at one particular target site in addition to multiple other target sites, but we'll just focus on one. We amplify each of those plants with a first round of PCR that holds adapters on the end. And then we follow that with a second round of PCR, kind of like a nested PCR, but it's a second round of PCR with primers that contain unique barcodes on the end. Okay, so each plant has an amplicon associated with a certain combination of barcodes, and that allows us to pool all of those PCR reactions into a single tube, clean it up, and then we send just that one tube for Illumina sequencing. And because we've added adapters for them, it's like $55. So we can assess the editing efficiency at a particular target site from in like 20 different plants, just as an example. You could do more or less. for about $55, and we can get a lot of sequence information. I average typically around 10,000 reads of a particular amplicon per plant. Okay. So we use a platform called HiTOM, which stands for High Throughput Tracking of Mutations. And that allows us to kind of efficiently and relatively cheaply look at the genome editing outcomes. But we do it all through a PCR and sequencing strategy to answer your question. So for that particular example, for 16 guides, we amplified all of those sites for each plant.
Okay, we have 2 more questions.
Awesome, thank you.
What are the challenges associated with the transfer more than 5 genes with their regulatory element into a heterologous host?
Some issues. Sure. So I don't have much experience actually in these kind of large multi-gene cassettes. I do a little bit, but what I will say in my experience, when I have multiple genes, I typically process them using a 2A peptide strategy. So I drive all of the genes together if you are interested in driving them with the same relative level of expression under, say, a strong polymerase 2 promoter like maize ubiquitin. I flank each gene with a T2A or P2A ribosome skipping peptide, and you drive it as a polycistronic mRNA. So I've driven over 5 different Cas genes like that, and that seems to work efficiently and effectively. But for other kind of gene stacking or multi-gene cassettes, I'm not too experienced in that, so I probably couldn't give you the best answer. But maybe you should look into, I know Roger Thilmony has a really nice system called Gauntry for assembling these very large cassettes using a recombinase strategy within Agrobacterium. So it's kind of in vitro recombination. And he can assemble tDNAs of tens of kilobases in size, which I've never done before. But I know.
Yeah. So usually, typically, the challenges the commonly encounter with the large gene cassette is the size of your tDNA.
Yeah.
Right. So if you can make your construct without difficulty, then you can use Agrobacterium to deliver the tDNA. But however, as you said, as the tDNA size get bigger, the chance of getting, delivering all the 5 cassettes at the same time gets lower. Like some plants only receives the first 2 cassettes or 3 cassettes, but missing the last one in the tDNA. So I think that's one of the bottleneck for many gene cassettes. But as Josh said, the gantry system is able to deliver over 10 kb tDNA into plants. That has been really validated. But the current limit is around like a 70 kb.
Which is huge still, right?
It is still huge. Okay, the last question we have probably, if we aim at knock-in via DSP-induced HDR, do you have experiences or suggestions regarding repair template vector design, specifically for increased copy number of repair template, efficient release of repair template, increasing knock-in efficiency, etc.? Thank you.
Yeah, I don't have a great answer for that because I full disclosure, I don't have any experience on doing gene targeting or knock-in. I know there are ways in which you can use viral replicons that people have used to kind of increase the level of templates available at the site of a double-strand break. I would point you towards that literature, but personally, I don't really have much experience in doing gene targeting, so I'd be afraid to comment much further on that. Sorry about that.
Yeah, it is really challenging. So, but you can also check Cortiva's paper about if you use morphogenic genes to stimulate cell division, you may increase the chance of HDR frequency too. Okay. Any additional questions? Okay, we have one more question. Is there any gene editing tool which can act like a disposable system? I think it can help to avoid any strict regulatory process.
Can act as a disposable system?
Disposable, like
Oh,
a transgene expression, I guess.
Yeah, so I'm not sure so much about the regulatory side. I know there are different systems that result in sort of I guess the cell death of any progeny that harbor transgenes. One of them is TKC, I think it's called, Transgene Killer Strategy by Yonde Zhao. You can look into that if you're interested in doing transgene-free work. However, yes, you can open your mic to explain your question. Sure. Yeah.
Hello, am I audible?
Yes.
Oh, thank you very much for allowing me to explain my question. Okay, actually, this is an idea. First of all, let me introduce myself. Myself, Shakil Ahmed, and I'm working as a biotechnology expert at the Seed Center facility of the Ministry of Environment, Water, and Agriculture of Saudi Arabia. This is a I don't know what's the progress is going on actually. I'm just a kid in this area, so my idea is, okay, we are facing several issues regarding the product regulatory process. If we are developing any gene editing product, some of the countries, they are lenient and they are comfortable with gene edited products. However, some countries are still considering them as just like as GMOs. So um, one day I was just thinking, okay, I don't know whether there is any system or not, but this is my question and idea as well. If there is not such type of systems, can we construct a vector or prepare a cassette and transform into a system? Whatever the function we want, whatever the aim we want to achieve, the goal we want to achieve, that construct, that system, do that, perform the editing, knock out or knock in the gene that we are going to target. Overall, that system perform the function and disappear. Just like we have a cup, disposable cup, we use it, we throw it.
Yes.
So
Yeah, we're actually running out of time, so I'll just try to quickly address your question.
yeah, thank you very much.
If you're using basic agrobacterium-mediated transformation, you will experience integration of the DNA, and if you want to get rid of it, you can either include, there's some recombinase strategies, like Crelox, for example, that allow you to excise the tDNA after the editing has taken place, albeit that will still leave a scar. Alternatively, you'll need to segregate out the tDNA in the following generations. Or you can use a DNA-free method such as RNP-based delivery. If you're doing CRISPR-based genome editing, right? You can deliver RNPs, no DNA will integrate into the genome, and that is likely regulated differently through your regulatory body. So in the interest of time, I can't really expand much more on that.
Yeah, we have only 50 seconds.
Yeah.
So let's answer the last question. Can we do knock-in over 2 genes and the knock-out over 1 gene simultaneously in sunflower?
I would say I don't know. That would be very challenging to do for a number of reasons. I mean, just one in particular off the top of my head, I don't work on sunflower at all. But if you're making multiple different double-strand breaks throughout the genome, and a repair template is already very unlikely to even recombine with one of them. But if you have multiple throughout the genome, you will experience likelihood of that template recombining at the wrong site, right? At the site you don't want to integrate, which could be a potential problem. But I think we have about 5 seconds. So I'll just thank everyone for listening to my responses.